Biology Part 189. The degree of curvature produced in the experiment proved to be an extremely sensitive biological test and bioassay for the presence of indoleactic acid, and was used to estimate the relative amounts of the auxin agar blocks after the coleoptile subjected to different treatments. For example, it was shown that two agar blocks separated by the thin sheets of mica would collect different amounts of the auxin if placed beneath the coleoptile illuminated from one side. The agar block from the shaded side contained more auxin and produced more curvature than the block of the illuminated side. The phototropism was therefore the result of an unequal distribution of auxin in the coleoptile, causing increased elongation of the cells on the shaded side. At first it was believed that the light caused breakdown of the auxin, but more recently it has been demonstrated that auxin is actively transported to the shade side. Consider the experiments illustrated, and in each case one end of the length of the coleoptile was placed in contact with an agar block containing indoleactic acid labeled with the radioactive isotope of the carbon. At the other end were two small agar blocks separated by mica sheet in an upright section of the coleoptile, placed in the dark amount of the labeled indoleactic acid detected in two blocks was equal, but in a coleoptile illuminated from one side or in one place, the horizontal position, the auxin was transported from one side to the other one and was distributed unevenly between the two blocks. Measurements of the total quantity of the radioactivity in the blocks indicated that auxin had not been destroyed by light. Although a number of synthetic substances have similar effects, indoleactic acid appears to be the only naturally occurring auxin, and its presence has been confirmed in many different species. It is produced from the amino and the tryptophan in the metabolically active tissues, including the apical meristems of the most shoots. It is normally transported downwards in the stem. Well, indoleactic and the acid promotes the release of H+ ions into the cell walls, reducing pH and providing the appropriate conditions for the activity of the enzymes which attack the crosslinks between the adjacent cellulose and molecules. The result is loosening of the cell wall, which allows the photoplast inside to absorb the water by osmosis, stretching and elongating cells. Precisely how the light stimulus is linked to the transport mechanism responsible for producing the unequal distribution of auxin, which causes growth curvature, is not fully understood. However, molecules of light-sensitive pigment concentrated in the cells nearest to the tip of the most shoots are likely to be involved. Negative protoplasm in roots. While the shoots of the plant are positively phototropic, the roots are normally negatively phototropic and tend to grow away from a source of light. The possible role of the auxin in the tropic of light response of the roots was investigated by exposing the root shoots different concentration of auxin. As we can see, that low concentration of auxin which stimulated the elongation in the roots had no effect on the shoots. However, the higher concentration stimulated the growth of the shoot but inhibited the elongation in the roots. Based on these observations, it was proposed that the negative protoplasm resulted from the redistribution of auxin to the shaded parts of the root as the shoot, but that the effect of the auxin were different. In shoot, high concentration of auxin caused the elongation in the cell's upward curvature, while the root in the high concentration auxin prevented elongation and therefore allowed the downward curvature. Geotropism. A geotropism is in the growth movement in the direction of determined by the gravity. Shoots grow vertically upwards and are described negatively geotropic, while roots grow downward and are positively geotropic. The importance of gravity in the geotropic response to the radicals of the broad bean seedling can easily be demonstrated by placing seedlings in a slowly revolving drum, a clinostat, as illustrated in the figure.